acids and obesity has been known for quite some time. It's the first paper published in the report from 1969, wherein obese individuals with insulin resistance, it was noted of the 20 amino acids that were measured, valine, leucine, and isoleucine, 3 branched-chain amino acids, as well as tyrosine and phenylalanine, 2 aromatic amino acids that share the same transporter, increased, whereas glycine is decreased compared to age and sex matched controls. And this observation kind of didn't have too many developments to explain it, until in 2009, my mentor at Duke, Chris Newgard, published a report using metabolomics where they profiled close to 200 metabolites and demonstrated among those metabolites measured in plasma of obese insulin-resistant humans, a statistical PCA factor that rose to the top, most strongly associated with insulin resistance when comparing obese and lean individuals, was actually made up of these same amino acids, 3 branched amino acids, valine, leucine, isoleucine, as well as their aromatic amino acids, tyrosine and phenylalanine, that seem to track with them, and some catabolic byproducts of branched amino acid metabolism. In the first step of branched amino acid metabolism, glutamate is produced from the transamination reaction, and the carbon skeletons actually can form acylcarnitines, C3 and C5 acylcarnitines, and these also showed up in that statistical factor, which strongly associated with insulin resistance. And so this kind of led a rejuvenated interest in understanding perturbations in branched-chain amino acid metabolism and obesity and how that might relate to metabolic disease, particularly insulin resistance. And I got particularly excited about this field of work when this second paper came out from Rob Gersten and Tommy Wang, looking in the Framingham cohort, where they found 12 years prior to diabetes development within the Framingham cohort, the same amino acids, leucine, isoleucine, and valine, as well as phenylalanine and tyrosine, were the metabolites most strongly associated with future diabetes progression. And so at that moment is when I reached out to Chris Newgard and said, can I come down and work with you? I'd love to kind of get some understanding about, this association and see if we can figure something out. And so when you think about this association, you think about branched-chain amino acids in plasma, you have to realize that there are many factors that contribute to the levels in circulation. And so it's really the product of their appearance from multiple different sites as well as their disposal. And so when we speak about appearance, of course, they're essential amino acids. We can't produce them ourselves, so they really come in the form of the diet. However, there is some information suggesting that branched-chain amino acids can be generated de novo by the gut microbiota. However, it's unclear at present how much the gut microbiota can really produce and contribute to the systemic pools. Although if you take an obese gut microbiota and put it into lean animals, you do see elevations of branched-chain amino acids. However, that is confounded by the fact that there are also metabolic disturbances that take place with that transplant, which may also contribute to the elevation of branched-chain amino acids. And then the balance of proteolysis versus protein synthesis will contribute to the amount of branched-chain amino acids in circulation. So in the context of insulin resistance and you have a shift in that balance, that, of course, can contribute to higher levels of branched-chain amino acids. And then in terms of disposal, of course, they can go into protein synthesis, as I just mentioned, but they can also be oxidized and broken down and even enter the TCA cycle as aneuploic substrates. Along the way in their catabolism, they can also produce bioactive intermediates. One that's well studied presently is called 3-Hib. It's derived from valine. And it's been identified to activate trans-endothelial fatty acid transport. There are also monomethyl branched-chain fatty acids. So some of the intermediates of branched-chain catabolism can actually be used by fatty acid synthase as a starting block to burning new fatty acids. And these have a unique structure. And within adipose tissue, these have been related to adipogenesis and playing a role in activating some of the transcriptional programs. So there are metabolites that are involved in signaling, and they can also contribute, like I mentioned, aneuploidically to the TCA cycle in the form of succinyl-CoA or acyl-CoA.
I'm sorry to interrupt. Can I ask a quick question? Terry Unterman here.
There are multiple tissues that then contribute to the oxidation of branched-chain amino acids, and muscle has been demonstrated, at least in mice, to play a major part due to the mass of the muscle. But other tissues of interest involve brown adipose tissue and also the liver. Liver is interesting because it doesn't possess the ability to convert this first step, which is where the branched amino acids are converted to their branched keto acids by the removal of the amine group, which forms that glutamate that showed up in that PCA cluster. Rather, they receive branched keto acids from other tissues that perform this first step, like muscle, that then go into circulation, and the liver takes them up and then performs this next step. And the next step is one I'll talk a lot about today, is performed by this enzyme BCKDH, or branched keto acid dehydrogenase. The enzyme complex is a lot like pyruvate dehydrogenase. In fact, it has many of the same components. And this is thought to be the rate-limiting step in branched chain keto acid metabolism, and the liver expresses a lot of BCKDH activity. Within the setting of obesity and insulin resistance, while muscle plays a predominant role systemically in catabolizing branched amino acids, there doesn't appear to be any modulation of the ability of muscle to oxidize branched amino acids, so that remains open. Rather, it is within the adipose tissue, the white adipose tissue, where there is a transcriptional downregulation of nearly every gene in the pathway, and also in liver, where there is a hyperphosphorylation of BCKDH that takes place that's thought to contribute to elevations of branched amino acids circulation. So when BCKH is phosphorylated by the kinase, an increase in the kinase BDK or a reduction in its phosphatase PPM1K, it becomes inhibited. And this is an inhibitory phosphorylation. And so the keto acids accumulate and there's reduced oxidation. And so this is really the setting that we have. In obesity, there is reduced insulin sensitivity, so reduction in the branched amino acids being put into protein, potentially increased input from diet, and then we have these metabolic disturbances at different tissue levels, which reduce the amount of branched amino acids being oxidized and catabolized, and that, I think, contributes to the increases in branched amino acids. But among them, which might be the causes or connecting features that link branched amino acids to insulin sensitivity and these other disease settings? And is it simply just higher levels of branched amino acids that explain the association? Are they the bad actors themselves? And so to set out to get at that, in the very early days, we performed some dietary restriction studies in fatty Zucker rats. And we used the fatty Zucker rat model. It's like the OB mouse. And the reason it's interesting is it, unlike a lean mouse model, doesn't need us to supplement branched amino acids in the diet to get these spontaneous elevation of branched amino acids. The insulin resistance itself, as well as the presence of these same modifications within the adipose branched amino acid metabolic pathway and the liver, are present within the fatty Zucker rat. So it mirrors at least the biology we see in humans. And so what you can see here is that in a fatty Zucker rat compared to a lean, there is an increase in branched amino acids within circulation. And by restricting the branched amino acids in the diet by 45% over 14 weeks, we're able to see normalized, almost normalized levels of branched amino acids. So the experiment intervention worked pretty well. So we performed hyperinsulinic euglycemic clamps in these animals to test whether that was sufficient to improve insulin sensitivity. And in fact, it was. We can see that there was a high glucose infusion rate required to maintain euglycemia in these rats. And we were able to trace the glucose into skeletal muscles. There was an improvement in muscle insulin sensitivity, here looking at glucose incorporation into skeletal muscle glycogen during the clamp. However, I'll have you note that this is a 10 milliunit per kilogram clamp, which is an enormous amount of insulin. In a lean, healthy animal, we're looking at 2.5 milliunit per kilogram clamps. And in those animals, you're looking at glucose infusion rates anywhere between 30 and 40. And here, with this massive insulin dose, we're still only raising up to about 13 milligrams per kilogram glucose infusion, which is quite small. So we didn't get these animals back to lean levels of insulin sensitivity by any means, although we did demonstrate that there was an impact of branched amino acids on muscle insulin sensitivity. And as I outlined there, if we do a glucose tolerance test in these animals, really this small increment in insulin sensitivity measured in the clamp setting really isn't having an effect in terms of a more physiologic maneuver where we give a glucose tolerance test and we see that glucose tolerance is equally impaired. And really there was no impact on liver metabolism in these animals. I'm just showing here that the inhibition of that rate-limiting step in branched chain oxidation remained inhibited in the branched-chain restricted animals. So BCKA is hyperphosphorated, stays phosphorylated, and so that impairment really isn't resolved by the dietary restriction. However, when we did look in the muscle, there were marked metabolic changes, and the most striking was actually when we looked at the acyl-CoA profiles. And what you can glean here is that many of the acyl-CoAs, including acetyl-CoA, the C2, all the way up to C10 and C12 acetyl-CoA, were drastically reduced. Even palmitoyl-CoA had a reduction in it. And the other thing that we noted was that the dietary restriction of branched-chain amino acids actually restored muscle glycine levels. And so you may recall from that first slide where they reported that branched-chain amino acids are up in obesity and glycine levels are down. This is kind of the first connection in the literature that we're aware of, which actually provided a potential explanation or connection or a hint about why glycine levels actually go down with obesity. And the fact that they were restored with BCAA restriction was quite interesting to us. And so the interesting thing about glycine is it's actually used as an efflux mechanism. There's an enzyme called glycine acyltransferase, and if you couple acyl-CoAs to glycine, you can actually excrete them into the circulation out through the kidney and through the urine. And what we identified here was with that restoration of glycine levels in muscle, there was actually a higher level of acylglycines in the urine, and acetylglycine being the most abundant one. We've heard about using carnitine for a similar mechanism, I think. And just to have you note kind of the mass that you can excrete here, there are 70 times more acylglycines in urine than there are acylcarnitine. So it's quite a predominant route for excretion. It's used a lot for excreting drug byproducts, et cetera. So we thought that was interesting. And we felt that the clue that branched-chain amino acids are influencing glycine was really striking. It might give us this new insight into where glycine was going in obesity. Why is it low? But glycine is actually used for many, many, many different biological pathways. I'm trying to illustrate that here with this slide. And so to go along each one and figure out if either of these are affected by branched-chain amino acids is going to take a lot of time. So rather than do that, we took the metabolite data and we ran a correlation matrix. And it turned out the metabolites that most strongly associated with glycine levels within the muscle of our animals were related to nitrogen trafficking. There was an inverse correlation between alanine and glycine, and alanine is, of course, involved in nitrogen disposal. And alpha-ketoglutarate, which is a nitrogen acceptor in this first reaction of branched-chain catabolism, is actually positive correlated with glycine. And the other thing that were correlated strongly with glycine was serine. And so we looked at these data for a little while and figured that if we coupled coupled that to the branched-chain metabolic pathway, you could see that this first step in branched-chain metabolism, the transaminase step, where the amine group is removed, is actually coupled to alpha-ketoglutarate to form glutamate. And what we know happens to glutamate within muscle is it needs to be exported to the liver to hit the urea cycle. And to do that, it's converted to alanine. And so we wondered whether nitrogen disposal could explain the depletion in glycogen, in glycine. How could that be? Well, to form alanine, when you have an increase in glutamate, to act as you need a carbon skeleton to act as a nitrogen acceptor. And the one that we use is actually pyruvate. And so you can make pyruvate from glycine by two enzymatic reactions. And so we hypothesized that maybe that with an increased branched-chain amino acid load going into muscle, that there would be increased transamination and there'd be a requirement for increased pyruvate generation to support the export of the nitrogen to the liver as in the form of alanine. These reactions had only really been demonstrated in the liver. So we performed some qPCR just to make sure the enzymes are also expressed in muscle. And in fact, they're expressed equally in muscle as they are in liver. And so we felt this was possible. So, excuse me. We went ahead and developed a tracing study. And so we performed a prime continuous infusion of M2 labeled glycine. And then took plasma samples throughout the infusion to look at downstream metabolites within this biochemical pathway. And we felt that if the label was found in serine, pyruvate, and alanine, that would be a demonstration that this indeed was a biochemical pathway that was taking place in vivo. And towards the end of the glycine infusion, we infused valine to see whether or not the addition of branched-chain amino acid load would alter the rate of this reaction. And so what I'm showing here is label enrichment during the infusion. And you can see that we got a pretty strong infusion labeling of the glycine pool within plasma, which stayed steady throughout the valine infusion. Then as early as 10 minutes into this infusion, you see the label being transitioned into the serine pool. And then, although diluted, there is labeling accumulating within pyruvate and also alanine. And the alanine enrichment dramatically increased as we started the valine infusion, adding the branched-chain amino acid load, suggesting that, in fact, yes, this is a biochemical pathway. So can you, excuse me, if this is true, if this is what's causing glycine depletion, then it stands that if you were to inhibit the transamination reaction in the branched-chain pathway, glycine levels should be restored. And so we were working with Lily at the time, who had developed a BCAT inhibitor, and I'm going to call LY for short here. And so LY will inhibit the BCAT reaction, which should prevent this pull on glycine as it's blunting the nitrogen disposal, or the need for nitrogen disposal. So here's in some lean Wistar rats, when we give the BCAT inhibitor, the branched-chain amino acids accumulate as you'd expect. Alanine levels go down, glutamate and glutamine go down. These are also nitrogen acceptor. So this is expected, and you see this small bump in glycine in circulation of these animals. However, if you do this in a fatty Zucker rat where there is branched-chain amino acid overload, here we see a much more dramatic increase in the levels of branched-chain amino acid in circulation, a much larger hit on the alanine glutamate pool. And now we see both glycine and serine levels rebounding quite strongly. And so we felt this gave us kind of a new explanation for why glycine is being depleted in obesity. It seems that the excessive branched-chain amino acid load is driving that.
Can you comment on the valine again?
This is really just a cartoon to summarize what we.
Can you comment? Can you comment on the valine again?
Amino acid metabolism, where the amine group is removed and forms glutamate. To facilitate the disposal of glutamate, we can break down glycine, and actually it'll release an amine group at the serine dehydrogenase step, which can be used to form glutamine, which can also be used for export, and also pyruvate, which can be used to export alanine. And by doing so, by depleting the glycine pool, we're actually depleting the pool available to the glycinase transferase enzyme to secrete acylglycines into the urine. GLIAD is expressed in muscle, but it's also expressed in liver. So it does, giving glycine back, actually really bolster our ability to excrete lipids. If you in fact give a high glycine diet to a fatty Zucker rat, you raise glycine by 150%, you do detect more acetylglycine and other short chain acyl group glycines within the urine, and circulating triglycerides will be lowered. So this is an area of research we're actively pursuing, whether we can modulate GLIAD levels within liver and impact things like NASH. I'm sorry. Yeah. All right. So that's kind of chapter one of the talk down. And so what I've told you a lot about is nitrogen handling, and how that pressure in muscle has an effect on lipid levels and has an effect on glycine levels, and how that might be a nice explanation for some of these phenomena. But what about glucose metabolism? And I've told you that just restricting branched chain amino acids, while it does all these things, really doesn't improve glucose tolerance. But when we looked at liver metabolic profiles, there was really no change, and this BCKDH inhibition was still present. So we wondered whether reversing that phosphorylation, that hyperphosphorylation of BCKDH in liver of obese animals, would elicit any other impact. And so we did this in two different ways. There was a small molecules that were designed to bind to the allosteric binding pocket on BDK, which would inhibit it. And the one I'm going to talk about today is BT2, but the first series was a compound called SCPP, and we performed the same experiments in those and got very similar data. And we also made an adenovirus to overexpress the phosphatase. So these are two orthogonal approaches to do basically the same thing, which is to strip the phosphate off BCKDH and reactivate branched chain keto acid metabolism within the liver. Okay? And we did that again in fatty Zucker rats. And these experiments were performed over 1 week. So they either got the compound daily for 1 week, or they had the adenovirus given to them, and 7 days later were sacrificed and stuff. Okay? And so at the top is the small molecule inhibitor of the kinase, BT2. And at the bottom here, I'm showing the adenovirus with the phosphatase, PPL1K. And I hope you'll glean from these data, as I've lined them up, that they gave very similar results. The phosphatase had a less potent effect on lowering branched chain amino acids than the small molecule inhibitor, which is acting systemically, which I think is to be expected. Both interventions had stronger impacts on the branched chain keto acids, which are the direct substrate for BCKDH, rather than the branched chain amino acids themselves. But what was really striking to us is that both interventions lowered liver triglyceride levels by 40 to 50% in this 1 week period without affecting food intake or weight gain. And if you look at those glucose tolerance curves, both the glucose and insulin excursions were lowered by both interventions in these fatty Zucker rats, where lowering branched chain amino acids themselves had no impact on this metric. And so,
Quick question.
metformin actually increases branched chain amino acid levels when you give it, because it drives phosphorylation of this step, which is interesting. But there's some papers demonstrating that metformin actually impairs branched chain amino acid metabolism across tissues. Yeah, metformin doesn't elicit this effect on branched chain amino acids. It does elicit about the same magnitude of effect in fatty Zuckers on glucose tolerance. So this is a pretty potent effect. And so these are very obese rats. They don't get the signal to stop eating, so they eat night and day, and they're incredibly insulin resistant. So this was a very potent effect that we were really excited to see. And we wondered how can it be that modulating this step elicits such potent effects on both glucose and lipid metabolism when lowering branched chain amino acids didn't? And so we felt that maybe the answer lies in phosphoproteomics. So we've inhibited a kinase and overexpressed a phosphatase. Are there other targets lying under the surface that we're unaware of? And so what we did is we performed phosphoproteomics in both studies and overlaid the data. And we felt that if there was something shared among the two, that would potentially explain how these effects were taking place. And so what we found was one phosphopeptide that was had reduced phosphorylation under both interventions outside of BCKDH. And it turned out it was the regulatory phosphosite on ATP-Citrate lyase. And ATP-Citrate lyase is the enzyme that takes citrate and converts it to oxaloacetate and acyl-CoA, and of course acyl-CoA being the building block for de novo lipogenesis, and oxaloacetate being a building block for gluconeogenesis. And by virtue of having these phosphoprotein lists, we were able to generate sequence motifs for the phosphosites, and there was a lot of homology across them. And what was interesting was that we determined that this site on ATP-Citrate lyase contained the known phosphomotif for the kinase, and also had an overlapping motif for the phosphatase, just like BCKDH. So we felt there was a strong chance that this could be direct. So if this is true, overexpressing the kinase itself in a lean animal might be sufficient to drive de novo lipogenesis. And in fact, that's the case. If you take an adenovirus encoded with BDK and you give it to a lean Wistar rat, you get an increase in phosphorylation on ATP-Citrate lyase on this same site. And we did this while giving D2O, and we're able to demonstrate that there's about a 2.5-fold increase in de novo lipogenesis in a lean rat simply by increasing BDK expression. Not only that, it turns out that BDK is also regulated by CHRBP. There is a CORI, or CHRBP regulatory element, within a enhancer region upstream of the BDK gene. Within liver of steatotic patients, there's a tight correlation between CHRBP beta and BDK expression. And if you fructose refeed to activate CHRBP, you get an increase in the known lipogenic genes, but also in BDK expression. And surprisingly, there's also suppression of the phosphatase. We still don't know how activating CHRBP suppresses the phosphatase, but giving glucose will do that. And overexpressing adenovirus containing CHRBP beta will do the same thing. So we wanted to know, does this effect rely on their ability to regulate the phosphorylation of BCKDH, or is it direct? And so we made some constructs lacking the mitochondrial targeting sequence. So this would not allow BDK to be within the mitochondria, but it would be within the cell. And in those cells transfected with the BDK that's excluded from the mitochondria, you got an increase in phosphorylation of ATP-Citrate lyase without an increase in phosphorylation of its mitochondrial target BCKDH. AKT is known to phosphorylate these sites on ATP-Citrate lyase, so we wanted to know whether that might be kind of downstream of BDK. And it turns out if you give an AKT inhibitor, you'll lower the background phosphorylation of ATP-Citrate lyase, but you still get the effect of overexpressing the kinase. And we went on to do some in situ experiments and demonstrate that, in fact, the kinase will directly phosphorylate ATP-Citrate lyase. So the summary here is that these enzymes that were known to regulate branched-chain amino acid metabolism actually play a role in regulating de novo lipogenesis. And so they, the disequilibrium between the kinase and phosphatase that happens with obesity and insulin resistance not only contributes to elevations in circulating branched-chain amino acids, but that those elevations are reporting on a system that is activating de novo lipogenesis or hypercharging it by phosphorylating this regulatory site in ATP-Citrate lyase. This is an active area of investigation in our lab. We're interested in how this is involved in NASH and NASH progression, but we're also interested in understanding, is this all that's happening to explain that 40% reduction in lipid? And so some new insights that we have that are unpublished at the moment is that SRBP1 also has regulatory elements within the promoter region and the enhancer region of BDK gene and correlates with BDK in human liver. And when we give the inhibitor for the kinase, in addition to lowering ATP-Citrate lyase phosphorylation, we see that it also lowers the expression of CD36 protein, the fatty acid transporter. So we think that this node is influencing de novo lipogenesis, but also fatty acid uptake. And that probably explains why you get these drastic effects on lipid levels within the liver within only 1 week. So we wondered if the branched-chain keto acids might be something that we'd use as a readout of active de novo lipogenesis or steatosis taking place in humans by looking at them in plasma. And what I'm showing you on this slide is that if you simply modulate the balance between the kinase and phosphatase within the liver, you'll get an elevation of the keto acids without an increase in the branched-chain amino acids. And so you get what you see is a shift in the branched-chain keto acid to BCAA ratio. And so we felt that could be a nice circulating index that you could use to look at NASH in humans. And so we got a cohort of individuals, bariatric surgery patients, BMIs over 40, who were discordant for steatosis and NASH. And we asked, among all the metabolites we can measure at the DMPI, whether any of them were associated with steatosis grade or NASH, and looked also at the keto acids individually and the BCKA to BCAA ratio. And long story short, of all the metabolite factors, there were very few that associated with either steatosis grade or NASH, but the only ones that associated with both was the keto acid from valine and the branched chain keto acid to BCAA ratio. And these withstood correction for HbA1c, body weight, age, sex, PNPLA3 variants, etc. We were able to get some liver samples from a smaller population from within this cohort and demonstrate that, in fact, the kinase mRNA correlates with features of NASH within human liver. And we're able to validate that using a Mexican obese bariatric surgery cohort, which was a little larger, working with Andrea Huertas Vasquez and Jake Lucis at UCLA. So it turns out that it seems that this node is an important regulator of de novo lipogenesis and lipid levels in liver, and may explain some associations of branched amino acids with metabolic disease. But what I've told you suggests that the keto acids themselves might not play any signaling function. They may just be bioreporters. And so I just want to share a little bit of data we've been doing in some of our cardiology studies, where we took isolated perfused hearts that were still beating in Langendorf preparations and perfused them with no keto acids, physiologic low keto acids are found in lean animals or levels that are found in obese insulin-resistant animal. And we performed a pure myosin infusion at the same time to look at protein translation. And what I hope you can glean here is that exposure of the heart to levels of keto acids that are found in obese animals, but not normal levels, is sufficient to raise protein translation by about twofold. So it's a very strong drive on protein translation. And we see massive changes in phosphopeptides with our phosphoproteomics, suggesting they also have a signaling function. So while I think a lot of the outcomes we see in terms of liver fat are related to other enzyme partners of BDK and PPM1K, the keto acids do seem to have effects on cardiac function. And so there may be this crosstalk between disturbances in liver metabolism and heart function, and these may play a role in that, and that's an area of active pursuit. So I've told you about lipid metabolism, I've told you about BCKA potential signaling, but I haven't gotten into our understanding about how glycemia and glucose tolerance is influenced by modulating this node. And this is still ongoing work, and we still don't have an answer for you. But we have made different models to kind of modulate the kinase and phosphatase in lean health animals to try and get some insight here. And so we performed overexpression study using AAV8 when our 8 weeks post-transfection with BDK, and in these lean, healthy CP7 Black 6 mice, that was sufficient to raise the branched chain keto acids and increase or impair glucose tolerance, increase the glucose excursion quite substantially. So lean, healthy mice, not more obese, just giving them more BDK, and you get glucose intolerance. So is this the keto acids doing this or BDK? And so one way we could get at that was to impair branched chain keto acid metabolism in muscle, which would then lead to elevations in keto acid in circulation and exposure to the heart, but not touch BDK or PPM1K levels in the heart. And so we did that by taking our PPM1K flox mice and crossing them with a doxycycline-inducible alpha-actin Cree mice. And so when we gave them doxycycline, we knocked down PPM1K within, tamoxifen, we knocked down PPM1K within the muscle, and we get these same elevations in keto acids in circulation as we got with BDK overexpression liver. So same level of metabolic impairment, systemically at least, but at a different site. However, in these mice, rather than having impaired glucose tolerance, they actually have improved glucose tolerance and a lowering of fasting glycemia. So deleting PPM1K from muscle, although it has the same effect on systemic levels of keto acids, does not have the same effect on glucose tolerance. To add to the confusion, we also have the liver-specific PPM1K knockouts, which have the same level of impairment in branched chain keto acid dehydrogenases within liver, the same level of increase in circulating keto acids, And these animals are characterized by low fasting glycemia, but not an improved glucose tolerance. So we have 3 different models of impairments in BCKDH that all lead to the same level of circulating BCKA, but each one of them has a different impact on glucose tolerance. So we think that at the moment, it's likely the branched-chain keto acids are not responsible for the glucose tolerance effects, and it's probably due to unique functions of the kinase and phosphatase in those different tissues. And we're doing a lot of phosphoproteomics to kind of figure out who else is involved in their signaling networks. And so the final project I'm going to tell you about is one in beta cells. So I've told you about peripheral tissues involving in glucose metabolism, but what about the islet? And so it turns out that PPM1K actually emerged in this systems genetics approach in 2012 as a gene that associates with multiple type 2 diabetes genes within the islet. It was one of only 14 genes that associate with more than, I think, 3 or 4 type 2 diabetes genes. And its expression within the human islets is associated with insulin secretion and HbA1c. It turns out, thanks to the TIGER database, which now has pancreatic islets alone isolated next to the GTEx data, it turns out that pancreatic islet is actually one of the sites of highest PPM1K expression systemically. And within human islets, we find that in type 2 diabetes, there's PPM1K is suppressed. So PPM1K is expressed in both alpha and beta cells within islets. So what is it doing in those cells? Is there a contribution of this network to beta cell function? And so we made the mouse promoter CRE, we cross them with our flox mice, and they're inducible. We give them tamoxifen, and then 4 weeks later, we've run a glucose tolerance test. And what you see here is that the PPM1K beta cell knockouts have improved glucose tolerance that's related to a stronger insulin secretion, suggesting that PPM1K is modulating something that's controlling insulin within the beta cell. If we go into INT1 cells in vitro and we knock down PPM1K, we get a very similar effect where we get an increase in glucose-stimulated insulin secretion with the PPM1K knockdown. But surprisingly, when you knock down PPM1K in beta cells, there's no effect on BCKDH, that first known target. There's no increase in phosphorylation as you might expect. And so we revisited our phosphoproteomics to look at potential other targets that might explain what's happening within beta cells when you knock down PPM1K. And one of the phosphoproteins that was most modulated in our liver study where we overexpressed PPM1K was the ribosomal protein S6 downstream of mTOR. And so we wondered whether PPM1K might modulate protein translation by acting as a downstream brake on this ribosomal protein. And so, of course, PPM1K by modulating branched-chain amino acid levels could alter mTOR upstream by altering exposure of leucine to the or it could be acting directly downstream. And so to figure that out, Jan Delay, a terrific postdoc in the lab, he performed Western blotting in these INT1 cells where PPM1K had been knocked down, looked at mTOR, S6 kinase, as well as S6. And while we don't see any changes in mTOR phosphorylation or P70S6 kinase upstream of S6, we do see an increase in phosphorylation on the major regulatory phosphosites on S6. P90 risk ERK signaling is also known to control phosphorylation on that site. We don't see any changes there. And in our cardiac-specific PPM1K knockouts, we see something very similar to what we're seeing in the INT1 cells, where knockout of PPM1K doesn't lead to an increase in BCKDH or changes in keto acids within the heart, but does lead to an increase in phosphorylation of S6. So we think that PPM1K might be an S6 phosphatase. And so if it's really downstream of mTOR, we should be able to inhibit mTOR signaling and still see these higher levels of S6 phosphorylation. In fact, that's the case, we can give a rapamycin acutely, and within 5 minutes, you get an inhibition of mTOR signaling to P76 kinase. However, the baseline higher phosphorylation of the cells that have PPL1K knockdown is preserved out to 20 minutes in these cells. If we inhibit mTOR longer by using ICAR to activate AMPK, we can go out 12 hours. Going out 12 hours, we have a pretty strong repression of mTOR signaling to P76 kinase. However, we still have this much higher phosphorylation of S6 in the PPL1K knockdown cells. So what's the consequence of that? Well, turns out, yes, indeed, knocking down PPL1K in beta cells, in addition to increasing S6 phosphorylation, increases protein translation measured by a pure myosin incorporation assay. You can see this pure myosin incorporated more highly across many of the proteins within the cells. And it turns out those cells, in fact, have more protein per cell and higher insulin content. So we think that what we're really getting is this drive on overall cellular protein translation when we remove PPL1K. That leads to having higher insulin content, and such when you have a glucose signal, more insulin release. So how could that go bad? Could that contribute to diabetes? Well, we know that too high levels of protein translation can activate an unfolded protein response. ER stress within beta cells has been linked to the pathogenesis of type 2 diabetes. And here we see that knocking down PPL1K in the beta cells elicits the hallmarks of an unfolded protein response. You have increased XPP1 splicing and higher expression of BIP. And when we go into islets of our PPL1K beta cell knockouts, we see already within a few days after knocking out PPL1K, an increase in XPP1 splicing. And actually, surprisingly, the gene that was most strongly changed was actually CHBB beta, which I had shown before regulates this node. And there's a suppression of CHBB beta when we knock out PPL1K. And so we wondered if that was connected to protein translation or something else altogether. And so Jan did a very careful experiment. He titrated cyclohexamide to turn down protein translation in our INT1 cells, and to match the knockdown PPL1K to the baseline levels of protein translation. So it didn't turn it off completely, but got it back down to baseline levels, and then looked at gene expression. So here you can see that he's lowered protein translation down to baseline levels, whereas in the controls, knockdown PPL1K suppresses CHBB beta expression. That's not the case in the cyclohexamide treated samples. And one of the targets of CHBB, TXNP, is actually involved in beta cell survival, is also, or beta cell death, is actually suppressed with PPL1K knockdown, and that's normalized when we inhibit protein translation. So this protein translation effect seems to be central to many of the impacts that PPL1K is having within the beta cell. And so studies are ongoing to see, under the setting of high-fat diet and beta cell stress, do mice lead to an accelerated form of diabetes when in the absence of PPL1K. And so our current working model is that within some cells, like the beta cell or the heart, PPL1K is actually having a strong effect on ribosomal protein S6, where its fluctuations act as to break protein translation by dephosphorating S6. And that may control normal levels of insulin production and secretion. Whereas in a setting where PPL1K is repressed, as in type 2 diabetes, or if glucose levels are high, and we've shown that through CHBP that suppresses PPL1K, that can lead to higher phosphorylation of S6, increased protein translation, and lead to states of ER stress, changes in gene expression through CHBP, and higher levels of insulin secretion, which in the early phase might be beneficial, but may be maladaptive also. And so simply to summarize, I hope I've been able to convince you that the metabolic impact of BDK and PPL1K extends beyond branched-chain amino acid oxidation. It's quite exciting. I'm really interested to see what we'll find next. And that although lowering branched-chain amino acids can have an impact on muscle insulin sensitivity, it seems like the kinase and phosphatase disbalance in the liver plays an important role in in controlling metabolic homeostasis. And so I'll stop there and thank the members of my lab. We've had many undergraduates and short-term trainees come through the lab at the moment. Our lab is made up of Jan, who did much of this work, and 3 research techs, as well as Rob, my compatriot. And we have a lot of important collaborators within the building. We do a lot of team science with Chris Newgard and the members of the Metabolism core and flux cores. Mark Herman, who used to be there, is now chief of endocrinology at Baylor, as well as collaborators at my alma mater in Quebec, where we get the human samples, and some collaborators elsewhere. Thank you. Yeah.
Just, I think on your last slide, you're talking about alteration of beta cells.
Yeah.
Can you repeat the question? It's hard to hear.
Yeah, so Brian just asked, on my last slide, there's a little arrow pointing to proliferation, and I didn't show the data today. We see that proliferation is slowed in these cells. So they're bigger, they have more the cells are bigger, they have more protein, but they don't proliferate as quickly. Yeah.
So there's Ernesto, who I was mentioning before, that's a model.
Yeah. Okay, terrific. So the comment there was to think about amino acid studies that Ernesto Bernal has performed. And so, yeah, and we are interested in understanding whether there's a synergy between having low PPL1K levels in the beta cell and having high branched-chain amino acid exposure as well.
But I also wonder if in the islet, image that beta cell features less ready makes that enhancement that you're seeing that maybe not real physiological change the beta cell structure.
So the comment was whether it's an increase in the cargo that's ready for versus a change in the actual function of the beta cells. And I think that's my take on it at the moment, and that's what the RO1 that's getting hopefully discussed is hopefully can dig into kind of defining kind of a lot of this. It's, you know, early days.
So I have a question and a comment.
Yeah.
And so first of all, really wonderful work, and really, really terrific to see how much you and your team have been able to learn about branched chain amino acids, you know, and all of the systems approach is really, you know, very beautiful. So congratulations to you and to Chris for hiring you or recruiting you, but, you know, to your team. Yeah, a couple thoughts, or one question, I guess, just to clarify, because going back to when, you know, Chris first found the the branched chain amino acid levels were high in obesity. You know, he thought, well, maybe it's coming from fat, but what you're saying, no, the problem is more in clearance and catabolism. And right? The levels are high because you're not clearing them. Okay.
I think it's complicated.
Okay.
Because there are so many different contributing factors.
Yeah.
I don't think we can rule out a major importance of insulin resistance causing branched amino acids to go up simply because you have reduced disposal and a shift in the balance of proteolysis. But there is underlying impairments also in the oxidative pathways, particularly in adipose tissue and also in liver, although muscle, there seems to be like a spillover model towards muscle, where muscle does a lot of it and it does even more in the setting of obesity.
But let me just clarify. So this defect in the the reduction in dehydrogenase activity would reduce clearance and utilization of branched chain amino acids, correct?
Yes.
Okay, so, at least inhibition of the dehydrogenase activity by itself might help to explain why levels are high.
Right.
Is that?
Yes.
And that's one of what I was thinking. And it might help to explain the initial observation that in the obese, insulin-resistant person, reduced dehydrogenase activity is important, not only for the effects on steatosis, through lipogenesis, but just in helping to understand why levels might be high, and that those two things are linked. I guess the other question, just from a kind of framing of what people have thought early on, when the idea of selective insulin resistance was raised. One suggestion was high branch chain amino acids elevate mTOR activity, which inhibits AKT, but still could promote lipogenesis. And I think, so I think what you're saying is that, a part of what you're saying is that the branch chain amino acids might contribute in insulin resistance through the mTOR pathway. Still, that might be part of what they do, but they're promoting lipogenesis not only through mTOR, but also through these other mechanisms that you're identifying. Or is that right? Or
right, yeah. So I guess the disturbance in their oxidation causes them to be elevated, while in parallel, the enzymes that are causing that disturbance are also activating
lipogenesis,
a mechanism to drive de novo lipogenesis and potentially influence fatty acid uptake in the liver.
Yeah, and
so
then
they may be a biomarker of altered biochemical pathways rather than direct actors, and they may synergize.
And do you still think that these, the effects on mTOR, which might affect protein synthesis as well in the heart, for example, do you think that's still a significant, target of the branch chain amino acids?
mTOR is a little tricky.
Okay.
I think you need the branch chain amino acids to prime mTOR for activation by growth factors, etc. But studies where we modulate branch chain amino acid availability, if you go and probe the mTOR pathway for signaling, often you see little to no effect on mTOR phosphorylation, unless you're looking in a situation where you've given a growth factor. So I know it's an easy one to add as a mechanism, but we don't, in my hands, in a lot of the studies I've done where I've looked at mTOR, I don't see it being modulated by the conditions that we're exploring.
Well, I think it's interesting just, that it was the first one people went to, because it was what they knew, and I think Brown and Goldstein suggested it in their, thoughts about selective insulin resistance, in part because they thought of it as driving SREBP1, so it's what they were familiar with. But clearly there's much more, involved and, really elegant biochemistry. I do have one other question, which, that I didn't get. And I did want to mention also that, more of a kind of a family history thing. My uncle was the head of biochemistry at Duke long ago, Phil Handler, who went on to become president of the National Academy of Science. And he got his PhD at the University of Illinois, coming from Brooklyn, in nutritional science, and went to Duke to do his research on amino acid metabolism. And, they really kind of, in a way, comes full circle to your work on glycine, and the centrality of nitrogen metabolism and its linkage to all of these other metabolic functions. So from a historical perspective, I think this is, in a way, part of a Duke tradition, if you will, playing a very important role in amino acid metabolism and its understanding. Having said that, could you, I didn't understand when you were talking about glycine, the effects of valine infusion or something, and how that affects the glycine story you were telling was linked to the branched chain amino acid story. Could you just clarify that?
Yeah. So the model that we propose is that the first step in branched chain amino acid metabolism, which is where the amine group is removed as a transamination step, requires disposal of that amine, or an ability to transfer it to molecules that can go to the liver for the urea cycle, and so alanine, for example.
Yeah.
And we felt that one way the cell could do that would be to take glycine, convert it to pyruvate, which then act as the amine acceptor to form alanine. So the tracing study was designed to look at whether, if we took a heavy labeled glycine where both carbons in glycine were labeled, could we pick up those carbons being passed along, so an N+2 shift in the components of that model. So of course we see a strong enrichment in serine, we see some carbons getting enriched in the pyruvate pool. Of course, pyruvate has a lot of other components that contribute to its pool, so there's a labeling dilution there. And then we see the two carbons showing up in alanine. At the end of that infusion, for the last hour, what we were hoping to do was to add a branched chain amino acid load on top of the system to see whether having more substrate would drive an increase in the conversion of glycine to alanine. And so that was what was happening at the end of that period. And what we saw was kind of a shift in the rate of appearance of a label within the alanine pool under those conditions.
Yes, thank you.
Suggests that it was sensitive to the branched chain amino acids.
Yeah, thank you. No, it's very, very helpful. Thank you. Very cool too. Thank you.
Thank you. Well, thank you very much, everybody.